Well, to switch gears a little bit in the Timm show, I go to the bright side of immunogenicity, and we have looked at the dark side, if I may say that, for 2 minutes, and I try to shift to the bright side, talking about tolerance, and this generation of antigen-specific tolerance. And you see through liver-targeted peptide-loaded nanoparticles, so this will be my favorite organ for the next half hour. Much more known in the metabolism field or sugar or glycogen storage or other fields, but it has a surprising role, we believe, to play in immunology in this. And let's see, this has been observed for a long, long time. So if one goes back and looks at old Nature articles, as shown here from '67, experiments have been done which gave a hint on the potential of the liver here. So here's a fairly easy hapten, dinitrochlorobenzene, which people have applied here to dogs. They got an antibody response. So one is control, two is just if you apply that subcutaneously. But then you gave the dog a chance to get it into the food, not poisoning them, but enough to obviously have a reaction in the liver before you subcutaneously inject that. And fair enough, the antibody level was quite significantly down. But if you circumvent the liver and don't give the dog the chance, although he's eating it, to pass it through the liver, you get the same antibody level back. So there seems to be a reaction. The liver is transferring and doing some good on the tolerance of this hapten. But there is more. 2 years later, another Nature article here where transplants were very much in hype and very much pursued, where between pigs, skin was transplanted in 3 different cases on the left-hand side, compared with the autograft. The picture is not that good from '69 and then reproduced, but it was rejected essentially. If you compare that with the autograft, none of these 3 trials were good enough to have a good transplantation. However, if you took 2 weeks before the liver of pig B into the liver or into pig A and then transferred skin from pig B, you can't distinguish it. So it's a full okay, and the transplant is fine, and the liver donor allograft looks essentially like the autograft. So that was surprising to those people, and speculation were about, but it was not understood. And actually, if I go to the next slide, this is more than 40 years later, so science takes time to understand what's going on, and here is the latest hypothesis which we are currently pursuing and building on. This is an excellent review from Percy Knolle and colleagues already 7 years ago, where he tried to dissect the immunology which takes place in the liver, which essentially has 3 players in that. Mostly the liver probably is known immunology-wise for their macrophages, which we call Kupfer cells. We do have some dendritic cells, liver-based dendritic cells as well. And then we have these little tiny cells here, which are liver sinusoidal endothelial cells, and they line the sinusoids in the liver, which are huge, they're very long, and have interesting functions, we believe. So Percy started as one of the first investigating these cells with their immunological properties and compared those to the Kupfer cells and to the dendritic cells. And then what we work on at Topas actually has been carved out by Ansgar Lohse and Johannes Herkel's group in Hamburg at the University Hospital, who spent even more time on these LSECs, like we call them, and how they compare to the Kupfer cells and to the dendritic cells. So they have features which we believe make them actually prone for playing a dominant role in the peripheral tolerance. So Percy compared, for example, the uptake of antigens in these 3 cell types. So LSECs, despite being endothelial cells, have great APC capabilities. They can present antigens really nicely. The import is 50 times more efficient than it is in macrophages, and certainly also in dendritic cells in this. But then they are, if you characterize them, have also the tool set to generate regulatory T cells, especially when it comes to the TGF-beta play in this. And I come to that in a minute. But also, this is not a minor population. Obviously, endothelial cells are everywhere in the body, but in the liver, the sinusoids are so large and so long that it makes actually quite a number of cells, and they constitute 15% of the liver cells. Keep in mind, they're also positioned right after the gut. That's not for nothing, we believe, when it comes to blood-borne antigens from the food. And they have great capabilities, not only in using the MHC2 pathway, but also in the MHC1 pathway. So they have a capability which actually Tregs like a lot. So when you do co-localization with the marker of the LSEX, where CD31 is one of these markers, together with the latency-associated peptide, one of the storage forms of TGF-beta, we do believe that LSEX can produce their own intrinsic pre-activated TGF-beta. But more than that, these glycoprotein A, which can bind this complex of pre-activated or of dormant, let me say, TGF-beta, which can come from other cells, it can store and bind that as well, just ready to use when T cells come by, naive T cells come by, to turn them into regulatory T cells. So, for example, Kupffer cells are quite capable of producing this TGF-beta complex, and LSEX are equipped with the GARP receptor just to keep it close, and when the naive T cells comes along and sees a certain MHC complex here, it is well prepared to activate that TGF-beta, which is required to turn the naive T cells into regulatory T cells. And that has been shown to produce those FOXP3 positive, CD25 positive T cells. Now, once they're there, do they do the job? Yes, they do. We work a lot on the background of multiple sclerosis and using the EIE model. So you see a number of pictures from that one. And what has been done here by Antonella Carambia and Johannes Herkel also is mix the equivalent effector and the regulatory T cells in certain ratios, and then look on the propagation of the effector T cells, but also in the interferon gamma secretion. And very clearly it can be shown and statistically significant that if you elevate that ratio of freshly produced regulatory T cells, you get what you want, you dampen the effector T cells, you keep them in check, you keep them back into the lymph node finally. So these regulatory T cells, keep in mind, they sit in the sinusoids, they are produced there, and then they go into the periphery and do their job at other sites, namely in the spleen and in lymph node and in other sites as well. So, LSEX. Jeremy, as much as I believe I'm a fox when it comes to the therapeutic goal of antigen-specific tolerance, I'm afraid I turn a little bit into a hedgehog when it comes to LSEX. So the goal we are coming from the therapeutic side is, if this is the division of what Percy is describing also, that the APC and the uptake and the immuno properties here are divided by size, LSEX take care of the smallest entities, of the peptides, of proteins, some nucleotides. Kupffer cells deal with bacteria, with erythrocytes as well. Dendritic cells in the liver are not so prominent, take even larger things up as well. Then, if we believe so much in LSEX, how do we address LSEX? How can we target those and deliver a cargo which is decisive to induce tolerance in this? And the solution is in the title as well, as much as we pursue it, and those are these nanoparticles. So the nanoparticles, very simply speaking, mimic blood-borne antigens. So blood-borne antigens mainly come probably from food, but they can also come from necrosis in the body, from scratches, from other sites. And those are processed, and there I come back that these L-sacs are just behind the gut, right, that they see whenever penetration of protein happens or peptides happen from the food, the 0.05% which are not digested, then they're the first they see that, and they take them up and process them. And we mimic with our nanoparticles essentially such a blood-borne antigen. It's very small. The nanoparticles is in the size of roughly an albumin, but it has to come with certain, mostly physical-chemical properties. So the size is important, the charge is important, the charge distribution and the zeta potential is important in this, in order to get the mimic right. This is the generation we currently work most on. So those are superparamagnetic iron oxide particles. For those who have been maybe in diagnostics, they call them SPIONs as an abbreviation and have been used also in the imaging diagnostic field as contrast imaging. They consist of an iron oxide core, and then they have to control the size also and polymer around that, which fixes the size of the particle, the inner core, but allows functionalization to the outside, which in our case is very useful. So we put the peptides to the outside of that nanoparticles, and in a fairly straightforward way to do that. And such a nanoparticle, which in the beginning has maybe a diameter of 8 to 10 nanometer, after decoration and polymerization is not more than 20 to 40 nanometer. And if you look at the last slide, you could see L-sacs are interested in everything which is up to 100 nanometer, roughly. And after that, then Kupfer cells come into play more and more. But one of these we can actually decorate with roughly 50 to 100 peptides, and that is by far not the capacity these nanoparticles could hold. But as we want to keep the physical-chemical properties for the targeting issue, that's good enough for us. So what do they do, and how do they interact with the L-sacs here? Here's an experiment from Johannes and Antonella and others in Hamburg, actually, where they have labeled the cargo and where they have labeled the core. And this is subjected to mice so far. The good news, first of all, was almost the same clearance curve. So no nibbling off of the cargo. There seems to be a corona around that, or attachment that any proteases or peptidases don't take off the cargo, which is very important. But then now you see L-sacs at work. In 15 minutes, 50% is gone. And in an hour, you have more than 95% cleared out of the plasma. And that's exactly what L-sacs should do. They should look for blood-borne antigens, and if they come by, they take them in, process them, cleave them, put them into the MHC2 pathway, and present, because that's their job. And that's what happens here as well. So it's cleared very rapidly in a very active fashion and ends up in the liver. Some goes into the spleen, probably also in some macrophages here, but that's it, essentially. So it's very rapidly taken out. But where are they? Well, you hear me saying it, they are mostly in the L-sacs. So this is a TEM into a sinusoid, which is covered here by the L-sacs at the rim. And if you home in, you see the particles actually, because they have an iron oxide core, very nice to watch at TEM and MRT. You see them inside the endosomes where they are processed. And our current hypothesis is really that we bandwagon a natural mechanism, and because natural blood-borne antigen are processed absolutely the same way, as you can see from fluorescent things, that they are put into endosomes which have a low pH, catepsins in there, cleaved into pieces, and then they fuse with the endosomes with the MHC2 complex, are loaded and presented to the outside. And we believe we do nothing else. So those are cleaved off, those peptides, or the cargo which we put in, is probably cleaved as well, and then we flood the MHC complexes which come by with our cargo of interest. And keep in mind, each particle has 50 to 100 peptides hundreds of particles are in one LSAC. So it's a competition process where we shy away all other natural blood-borne antigens and just load the MHC with our peptide of interest. Now it comes out, and then naive T cells come by and are turned into regulatory T cells. And one more piece of information to understand liver immunology: sinusoids are so abandoned in there, the bloodstream really gets low in the liver. Although, portal vein is really strong when it comes to sinusoids, they fade out and are very slow flow. And naive T cells stay 30 minutes in such a liver. That's the organ where the interaction of naive T cells with an organ is the longest in the whole body. So there's ample opportunity for interaction and communication between these naive T cells and the LSACs, which stream the whole sinusoid from this. And that, we believe, happens. So mechanistically, we believe we are backriding a natural process here. What are the effects? Well, as I said, for logistical reasons, for grant reasons, for other reasons, we work a lot on the background of multiple sclerosis. So in this way, the group took different EAE models here, which are probably known to all of you, but we try to use different genetic background, use different epitopes, one from the MVP, one from the mock here. This is already a 20 mer or 21 mer. And those were loaded onto those nanoparticles and subjected one day after immunization into these EAE mice. We still call that preventive. So you see the PBS control, the unloaded nanoparticles, and then the nanoparticles which are loaded with the peptide of interest in this. And that's quite a radical reaction. That's a one-time application of these nanoparticles one day after immunization, and we couldn't see any EAE anymore. This shows 45 days. We have now data for 70 days, same picture. We have not re-challenged yet. We need to do that, but so far it looks very impressive that we have generated a population of regulatory T cells, because those are our workhorses, not so much the nanoparticles. The nanoparticles are inducers, but the workhorses are the regulatory T cells, essentially, which seem to do a good job here. Does that work in a therapeutics setting? We believe yes, in the first go, right here. So we waited until the EAE mice had a score of 2 or higher, and then gave a one-time injection. So this is 10 days after immunization. And we get it down to below 1, which then is hard to distinguish if the mouse is affected or not. So that's a promising approach, although our therapeutic setting will, in MS, be between rashes. It's good to see that even in a therapeutic setting, we get a very clear effect of this methodology. So, why we believe this is a nice approach also in the therapeutic sector using these particles. We can trace them. As said, they are an oxide core. It's beneficial to see where they are. Regulatory offices in Germany already applauded that because they can always see where it is in the patient. That's very beneficial. We can dose it, I think, like hardly anybody else, and that's again due to my beloved LSACs, because they have this active uptake mechanism and are so strong that we can pharmacokinetically really adjust the dose we're giving because we know where it ends up. It is biodegradable, the code we're using. We spend a lot of time now to see and follow the kinetics of where it goes and what it does. Keep in mind that if this works, then application of such nanoparticles is probably foreseen every 6 to 12 months, because the workhorses are the T-regs, not the nanoparticles. And T-regs live between 100 and 300 days, so we need to re-stimulate. That is true. But they have a good, so we will have a good window in order to make the particles disappear before we come next. And then we're currently spending a lot of transferring the originally academic protocol into a production process, which obviously, for some of you, you will know that you need GMP certified equipment to make this, the analytics and all that stuff to make real nanoparticles as therapeutic out of that. When it comes to regulation, it's actually good that some of these biomes have already been approved by the FDA. So here's a little bit of history where this stuff has been gone through a regulation process, and it's actually amazing to see I saw the hints of FDA for immunogenicity. They are clearly there since some time. But even for nanoparticles, you go to EMA on the website or the FDA, and they have very clear regulation what is needed to get approval. And this helps us, the protocols, how these folks did it, helps us to truly get to a product. We will not fall short on applications. There I come to Jeremy in a minute. So this is from my colleague John, who spent half an hour to come up with 100 autoimmune diseases. And there are probably more than that. And you can break them down in various applications. Obviously, we do need to know what the antigen is, or the antigens are. Even better, we need to know what the peptides are. And we actually, in this case, do love peptides, because peptides also have a stoichiometric part to play in this. If we only would take the protein, we may have, in this competition arrangement, by getting onto the MHC complex, a problem. But if we have peptides and load them 50 on each nanoparticles, we just flood the system. So ideally, we need to know the antigen or the antigens and the epitopes on this. But it can be more than one, because we can mix these nanoparticles who are decorated by different peptides. To get to the right epitopes, we go to Jeremy, for example, and find out what are the true ones which come from human dendritic cells, for example. But this is only covering the autoimmune space here. Obviously, if this principle holds, then allergies are something which we also will tackle as long as the antigen is known and we know where the epitopes are, and certainly also the ADA complex, in case we know where the antigenicity is caused from in these proteins. Okay, we tried that with a team, a very small team at the beginning. We have received some financing as a biotech company, and have really good advisors at hand. Percy has also committed in this, and advisors who have done nanoparticles. Here you see a chemist stumbling into the immunology and is fascinated of what even the liver can do. But I'm happy to take questions, and I'm happy to have Sabine with me, who is in the audience as well, who knows much more about immunology. But I hope I could get across it's sometimes worse to be a hedgehog, Jeremy. Thank you very much.